Lena moved here from Chicago, Illinois, after working in biomedical research for the past 3 years. She's interested in translating those skills to conservation genetics, and you'll get a little taste of that today. Since moving here, she has especially enjoyed avoiding the long Midwestern winters, riding on the back of Jorge's motorcycle, and soaking up the year-round sun. The title of Lena's presentation today is Quantifying the Unseen, eDNA Insights into the Enigmatic World of Sea Turtle Genetics.
Thank you, Samantha. Thanks for being here. My dad's already crying. Yeah, so if you heard the introduction and my project title, and the only words that really stood out were sea and turtle, you probably imagine my project looks something like this. Having always felt a strong connection with animals, I aspired to work in wildlife conservation. Being outside, exploring nature was how I wanted to be spending my days. I chose my undergraduate program based on a brochure of students holding baby black bears for a class, which I ironically never took. Instead, I became fascinated with microbiology and genetic diseases, leading me to work in various labs and contribute to several papers on cancer genetics. Being a part of this program now has felt like coming full circle, merging both my passion for wildlife and scientific research. And of course, getting to work with sea turtles is an added bonus. But while being in the field is undeniably a crucial part of research, by just looking at the surface of the ocean, you can only see a fraction of what lies beneath, if anything at all. But what if we could capture the biodiversity of the ocean just by filling up a bottle with seawater? It turns out you can. Because all living things leave their DNA behind by saliva, feces, urine, or skin cells, like a trail of breadcrumbs, we can pick up samples of water, soil, air, etc., and see what's in that area or has recently passed through. This is what we call eDNA. eDNA is revolutionizing conservation by allowing for the detection and monitoring of species in a non-invasive and cost-effective way, making it an ideal tool for protecting wildlife and their habitats, especially those that are highly mobile and hard to locate, like sea turtles. And not only are they hard to spot, spending up to 99% of their time of their lives in the ocean, but all 7 species of sea turtles are listed as vulnerable to critically endangered. eDNA technologies offer a promising solution by allowing us to detect and analyze these species without ever having to see or capture them. Thanks to this preliminary study, we already know you can scoop up water next to a sea turtle and detect its DNA. But while detection of eDNA is useful for confirming the presence of sea turtles in different locations, my project's objective was to take this one step further by quantifying eDNA. Quantification could provide much richer data that are essential for detailed population assessments, monitoring changes over time, understanding habitat use, and informing effective conservation and management strategies. In South San Diego Bay, there's a green turtle hotspot that's been monitored by NOAA for 3 decades, with the first records of foraging turtles dating back to the 1800s. These green turtles congregate especially in this small inlet, making it an ideal site to sample because we're able to spot and count heads as these turtles come up to breathe. For a baseline study like this, it's really helpful to have a group size estimate by comparing the number of turtles that we see to the quantification of the turtle eDNA. This way we can create a baseline understanding for future projects. The water samples were taken in a dispersed pattern to get a good full coverage snapshot of the site as a whole. Okay, so we brought the collection bottles from the lab in a cooler and transferred them into a boat to head out to the turtle dense area. We collected a total of 59 samples from 4 different field days. The samples were collected right at the surface of the water, and the bottles were kept in the cooler with ice packs until we got back to the lab. Back at NOAA, I'm working in a lab that's specially designed for working with eDNA samples. The biggest worry is contamination, so everything must be cleaned. The water samples are pulled through a filter by a vacuum system, leaving any DNA picked up from the water trapped on top of the filter. Each sample of 2 liters takes about an hour to filter, so as you can imagine, I spent a lot of hours watching water move very slowly. This 20-second video is actually showing 75 minutes of work. Once the filtering is complete, the filters get rolled up and stored in a buffer that allows for the DNA to fall off and mix in with the liquid. But to make sure that all the DNA gets off of the filter, we move into DNA extraction. The goal of this 2-day procedure is to purify the DNA from the filter because the filter also captures a lot of junk that we don't need, like sand or dirt from the bay. Both days of this procedure take around 3 hours, so the bulk of my project was really spent filtering and extracting these samples. If you enjoy lab ASMR, I've got hours and hours of real-time footage that I'm happy to share. So now we've moved from 2-liter bottles of seawater to just a few drops of pure DNA, but this could be DNA from any species in San Diego Bay where the water was collected. In order to find out how much turtle DNA was in the sample, we can use droplet digital PCR. This cutting-edge technology has never been used before to quantify sea turtle DNA, eDNA, and the machinery has not been used yet at NOAA, so part of my project was also getting it up and running for everyone to use. So what is ddPCR? ddPCR is a process, and the first thing that it does is take each sample, a single drop of solution containing DNA from various species, and splits it up into around 20,000 even smaller droplets, each of which contain varying amounts of DNA from different species. Because each droplet is much less complex than the sample as a whole, this allows us to locate and target the DNA much easier. And what's locating that target are specially designed tools called primers that act as magnets and only attach to the turtle DNA. Once attached, the primers then create billions of copies, essentially outnumbering any other DNA present in the solution by a process called polymerase chain reaction. Another tool called a probe is also specially designed to attach to the target and fluoresce, acting as a highlighter that will light up all of the copies of the target DNA. So what we're left with after droplet digital PCR is that only the droplets that contain the target are going to fluoresce, and these will be counted as positive droplets. And by comparing the ratio of positive to negative droplets, we can then quantify the amount of turtle DNA that was in the original sample. So how did this work out for my water samples? Before actually getting to run the ddPCR, I had to spend a lot of time designing the tools, the primers and probe, that would reliably stick to and highlight green turtle DNA. And for sake of time, I'm just going to touch on the design briefly. These tools were created to recognize a small sequence of the green turtle mitochondrial DNA within the CO1 gene. This gene is commonly used for identification because it's highly similar across species, but different enough that we're able to tell one from the other. And this may look scary, but all I wanted to show here is that the sequence of the probe was designed to perfectly match to the green turtle DNA sequence. This is where the probe will attach and light up the sequence wherever it is found in my samples. Okay, moving into the results. The first thing I did was make sure the primers and probe worked using pure green turtle DNA as a positive control. I used this DNA extracted from the skin of a green turtle and diluted it many times to see how well my tools could find the target. This helps me to determine a lower limit of detection for my environmental samples because the turtle DNA will be much more dilute in a sample of seawater than directly from the turtle's skin. To read these results, each dot represents one of the 20,000 droplets, and the samples are split into, each sample is a different rectangle moving left to right. The blue dots are positive, meaning they contain the target DNA, and the gray are negative. The higher up the dot is on the y-axis means the more copies of the target there were within the droplet, so essentially they fluoresced brighter. And moving left to right, the samples are more and more dilute. So these results are just showing that the primers and probe can indeed detect green turtle DNA, even up to diluting it 64,000 times. These are negative controls, just water, no DNA, to make sure that there's no error in the machine or contamination on my part, so no detection there is exactly what we want to see. And to see if the probe is only picking up turtle DNA and nothing else, we ran a few fish to check for specificity. So again, what we want to see is no positive droplets. So this one's great. Perfect again, nothing could go wrong. And this one did not work. But what happened? So that sample was an OPA, and looking at the probe compared to its DNA sequence, even though there were a few differences, meaning the probe was not a perfect match, it still attached to and lit up the OPA DNA. This is ultimately not an issue, as we can send out these samples for sequencing, which will tell us what species the DNA belongs to, and there aren't going to be any OPA in San Diego Bay, as they're a deep water fish. So finally, with the water samples, we were able to determine that the design does indeed quantify eDNA in San Diego Bay. Again, to make sure this DNA belongs to green sea turtles and nothing else, we can sequence the samples for proof. But these results are exciting, and this technique can now be readily used in future assessments of San Diego Bay and other NOAA study sites. For now, it can be used together with field research techniques like counting heads and capturing individuals, and can continue to be optimized for monitoring relative abundance and informing management and policy in the future. Perhaps even someday replacing the need for the other more difficult and invasive methods that have been relied on so far. This project will continue to move forward by running more of the environmental samples, since I was only able to get to a few by the time I was done filtering and extracting all of them. We can sequence the samples and continue to optimize the design of the primers and probes. One way that we can minimize crossover between species with the probe is to target a less conserved region of the mitochondrial DNA, such as within the D loop that would be more specific to turtles. The more immediate deliverables from this project, aside from the design of the primers and probe, are standard operating procedures for eDNA extractions, as well as for the droplet digital PCR. These can be used by other labs at NOAA also interested in quantifying eDNA of marine mammals and fish, and I know there are other projects already in the works. There are, of course, a few kinks to be worked out, but ultimately eDNA could completely change how we find and monitor all sorts of living things. And with increasing threats to thousands of species, the unique insights we're getting from eDNA are more important than ever. Although I didn't need to see any turtles in order to complete my project, it was really helpful that we had such a great site right here in San Diego Bay to establish a baseline. And I was lucky enough to see these amazing creatures up close, as I'm only a small piece of all the work going on at NOAA to study and protect these species. While it may seem like a small step forward in terms of sea turtle conservation, I find this work incredibly exciting and promising for many future studies to build off of, with environmental DNA really just starting to pick up momentum. Ultimately, nothing can move forward without foundational science like this, even if it moves at a turtle's pace. Thank you. And thank you. And thank you to my capstone committee as well, and everybody at NOAA who helped me make this project possible, especially Amy, Erin, Sue, Vicky, and Brittany. And that's it. Yes.
Actually, do you know, there was a moment early on where you identified, I think you called it a probe, that would help you have it light up. How did you know what that substance should be? I mean, how do you choose the right medium for the probe?
So the probe itself is like a short sequence of DNA. So it's basically as long as I can pull up, and there's an online database called GenBank, which has all like the genomes of many species. So as long as I can pull up that and check and see that it matches perfectly, like a small short piece of the sea turtle mitochondrial genome in the CO1 gene that I'm targeting, that's how you choose it. Yeah.
All right, I've got a question. Thanks for that presentation, Lena. Will this technique be used in future field surveys at NOAA or by other departments at NOAA?
Yeah, so right now there's like the turtle department at NOAA and a marine mammals department as well, and they're already in the process of designing and ordering their probes. So now that the ddPCR is up and working, there's lots of projects that are going to be using it now, and it's already been used for a few other things as well. It's really cool.
I have one more question. So what are the immediate deliverables of your project? What will happen next?
Yeah, so basically it's just the standard operating procedures that I designed, as well as the design of the primers and probe. So that will continue to be used by for other projects, and the SOPs that were designed then can be used now for any environmental DNA extractions and anybody who wants to use the ddPCR.
Great. Thank you.